Hey, welcome back, or I mean, I should say, I'm welcome back. That's right. Welcome back. Ooh, welcome back. Welcome back. Welcome back, Carter. I don't know what's going on here. This tube should not be here. It should be on this side. There we go. Okay. We're going to do terminology today, and we will start with a review of biology for our evolution and ecology class. And so we're going to talk about things that deal with inheritance on a micro scale. And on a micro scale, that has to do with mutations, genes, and your alleles for the individual organism. Now we can look at an organism's chromosomes, and we can look at them fairly easily in a picture. And so we can see the karyotype, which is the visual representation of those chromosomes of that individual in a picture. And that really is important. Especially if we're looking at that genetic information, because we want to establish some kind of family pedigree. It may be important to trace the lineage of a family, and that's what it does. It traces the lineages of members of a family, and you can trace individuals all the way back. You can look at successes and failures in each one of those individuals and determine if any of that is going to be passed on to its progeny. And so there's some terms we need to go over about chromosomes, and first we're going to talk about linkage. Okay? Linkage is the connection between the chromosomes. And sometimes unfortunately you get non-disjunction where they don't separate during prophase, metaphase, right? P on the mat. Prophase, metaphase, anaphase, telophase, okay? For mitosis. If you're talking about meiosis, don't forget you have to go prophase, anaphase, metaphase 1, and then you have 2 before you have telophase again, and then cytokinesis. Well, actually, you could have cytokinesis in both of those steps. You can. All right. So non-disjunction, right? They don't separate, and that's a problem. Down syndromes, for example, right? Trisomy has more chromosomes than necessary. Usually duplications of that, right? Which is not duplication, but having more than the required amount is harmful for that organism, and the organism usually doesn't survive. How about another one? Let's talk about duplication. Well, let's say for some reason during that life of a cell, region of a gene or something gets duplicated twice, right? You can have 2 of them. You have deletion. You can lose areas of genetic material, or you can just lose one. And you have a point mutation that can just be one. You can lose whole sections. As I say, you never know. Some of them you never know if it's beneficial or not because you don't see those organisms because they never get reproduced. Alright, so we covered karyotype, we covered linkage. Karyotype, remember, that's going to be the picture, the detailed picture of the chromosomes of an organism. We're going to see linkage. And linkage, you know, is the chromosomes as they come together. I'll make that. They kind of look like little X's, right? The pedigree, the family pedigree. It can trace the lineages of related family members. And in order to do that, you can track back diseases, and then we have deletion, and then inversion. So inversion is where a section of the DNA, or maybe even a whole allele, gets reversed. So a sequence of A, T, C's, and G's gets reversed into G, C's, T's, and A's. And that's an inversion. Translocation is where a region of DNA gets cut out of one chromosome and inserted into another, totally different chromosome. And we talked about non-disjunction, which is the failure of the chromosomes to separate during replication, right? So during mitosis. And usually that's going to cause death of the organism. If not, then it can translate into diseases that we see, like trisomy or Down syndrome. Trisomy 21. All right.